I didn't put my name up here, but I'm a professor at Auburn. It's a big state school in Alabama. And instead of telling you really about myself, I thought I'd just sort of explain where I think I fit in the group, because I really don't study mannequins. I had a student study mannequins for a while, and I've seen a few, but I'm not a mannequin expert by any means. But I've always been really interested in sexual selection, and mannequins are sort of the poster child for sexual selection. That's the point of our group, really. And so I've been thinking about sexual selection for a long time, since Rick Crum and I were sitting in an office in Michigan as grad students together. And recently, I feel like I've gained a bit of clarity about what's shaping female mate choice patterns, or maybe just the fog of old ages. Everything seems right. But I recently argued in a paper that there's 3 fundamental classes of information that any female assessing a potential mate should be looking at. And that is the condition of the male, and we can talk about what condition means, the species identity of the male. You don't want to mate outside of population boundaries or species boundaries. It's usually a really bad thing. And then the aesthetic beauty of the male, the sexiness, which is a really important part. And they're in no way competing with each other. Every female needs to know about each of those classes of information in prospective mates. And so I've spent a lot of my career focusing on one aspect of this, which is signaling condition, and how that would work, and especially in birds, and especially in pigment systems, so carotenoid pigment systems. So when we're talking about especially red and yellow coloration in mannequins, or basically any bird, the basis for that coloration, with very few exceptions, is carotenoid pigments. And this is the key pathway that dictates whether a bird basically is going to be yellow or red. Whether a bird expresses its carotenoids as yellow, which are almost the universal dietary pigments, or is red, depends on whether it uses an enzyme to biochemically modify those pigments from yellow to red. So this is a ketolase. This is an unknown enzyme, but I've been working with a group of geneticists and physiologists to find this gene. I think we're on the cusp of finding this gene, and I think with the genomic resources that come out of this group, we could really make some headway using these new genetic discoveries in color to understand the basis for color variation.